Well, it is my great pleasure to introduce Mike MacCoss, who I think doesn't need an introduction by that point, but let me still say that Mike is really a very, very influential figure in mass spectrometry-based proteomics in general, and in computational mass spectrometry in particular. Mike was the person who was at the origin of Skyline together with Brenda and made this project happen and is continuing to make this project happen. And well, among many other accomplishments, of course, DIA was really the type of data and the type of problem where Mike has a tremendous impact. And I'm really thrilled that Mike, we were just talking about how much travel he has, and I'm really thrilled that Mike made his time to be with us today here. Thank you so much.
Okay, so I want to kind of give you a little bit of thoughts, like how we think a little bit about data independent acquisition and how we think about it in the context of doing quantitative proteomics. And as Brian kind of mentioned, kind of our lab's gone all in for this. I think you know, and I'll also point out too, we started doing data independent acquisition in about 2004. We collected this data in our lab, and it took a lot of time for both the hardware and kind of the ideas and the methodology to kind of catch up with this. But we definitely were collecting data on unit resolution linear ion traps back in 2004. That was data independent acquisition. In fact, I've just started an email exchange with John Venable, who wrote the first ever paper on data independent acquisition, and he's now starting to get back into it too. So it's kind of interesting how these things kind of come around at times. So I do want to give a little bit of some definitions. So the first thing I want to get a definition is the term identification. It's thrown around a lot. I will kind of define identification as the action of proving an item's identity. So identification, you can identify the peptide that contributes to this sequence, this spectrum. But it's hard to say that you've identified albumin in plasma, given the fact that albumin was identified in plasma back in 1924, and actually the full amino acid sequence was derived in 1975, right? So a lot of people say they ID something, and a lot of this stuff is a carried over term from the old idea of you know, collecting data dependent acquisition and defining that a certain thing is determining the identity of those individual spectra. So our lab actually tries to use quite frequently the word detection, because what we're more interested in, these things that we know are in the sample, can we detect them? Are they detectable? So that's something that's a little bit, we're not trying to define something new that hasn't been discovered before. We're trying to determine whether or not that thing is detectable in that sample. The other term is this term quantification, because it's also thrown around a lot in proteomics. And I will go as far as saying quantification is a measurement where a change in the signal reflects a change in quantity. Now, I'll point out that I don't think that this is true for a lot of quantitative proteomics experiments. There's a lot of cases where people find that two signals are different from one another, but they don't necessarily define that the change in the signal that's measured is actually reflective of that change in the quantity. So then what I'll do, because of these definitions, I'll add in another term, which is this idea of differential measurement. And that is that where a measured signal is different between two conditions, but the magnitude of the difference might not be quantitative. People have any questions about this? This is kind of like a little bit different way of thinking about this, but this is definitely the way we try to define some of these terms. So I'll argue that a lot of quantitative proteomics are actually differential measurements, and that a lot of cases where people use the term that they identified something in a mixture where actually they just detected it in that sample. Okay. So anyway, it's something that I think that we kind of have to sort of, as a field, begin to think a little bit about terminology. So the other thing I struggle with, I came from a small molecule quant background. And in there, you would often go through this process of building an assay. You build an assay to measure an analyte or a set of analytes. But what is a proteomics assay? So generally, when you develop an assay, it involves defining the analytes that are being measured. Is the assay measuring what you think you're measuring? So is it specific? What are the figures of merit of that assay? So does a change in signal reflect a change in quantity? Do you know the limit of quantitation? Defining the limit of detection makes the absence of signal often meaningful. So you know that there's a limit at which you can detect something. So if you don't detect it, it must be below that limit. And then what is the within batch and between batch variance? So often now samples sets are being done in hundreds and even thousands of samples. And if you can only prep a certain number of samples, what is your within batch prep variance for the samples, and what's your between batch set of variance between samples? And then there's signal calibration, which is can I take data that was collected in one lab and compare those signal measurements from data collected in another lab? Can I take data that was measured on a triple top and compare those intensities between data on a Q exactive? That's the goal of signal calibration, and I'll talk a little bit about ways in which we think about this as well. So why is this sort of assay validation important? So a key component of it is it's essential to reproducible research. How often is it that you get to see where someone does an experiment in, let's say, in one person's lab, and they can reproduce that exact same results in another lab? It's fairly rare, and proteomics won't advance to being to a higher level of a standard of a field until we get to the point where we have an assay that's reproducible between different labs and different groups. So reporting a measurement that others can't reproduce is detrimental to the community. So just being the one lab that's capable of doing something that's unique and special is not very helpful. So it also enables us to put a price on output. So genomics put a price per sequence base initially. It used to be the cost per base. Now, of course, it's now a price per genome. But this became a target for the federal funding agencies to start to drive innovation, right? So what sort of things could be done to drop that cost? So what are the things that we could do to drop the cost of proteomics? Do we even know what the cost of output, information output is for our experiments? And then you can start to think about costs could be improved in two ways. You could either think about reducing the time while maintaining the multiplexing, like the number of analytes being measured and certain analytical figures of merit, or you could actually increase the degree of multiplexing while keeping the assay length and figures of merit the same. So you could measure more analytes per unit time, or you could measure the same number of analytes per unit time but shrink the time, right? So that's definitely two ways that you can think about innovation. But we don't normally think about this right now. We're so interested in improving the size of our lists that we often ignore the fact that, okay, well, what were our figures of merit, except for the fact that we got a certain number of peptides that were in our list. Okay, so what do we need? So I think we need methods that sample the same peptides in all samples. That's kind of why we're talking about data-independent acquisition. We'll talk more about that, where we talked a lot about it. We need methods where the change in signal reflects the change in quantity. So we need quantitative measurements, not just differential measurements. We need methods where we can get the same signal despite differences in sample preparation, instrument platform, etc., so calibration. We need to recognize that changes can occur with individual peptides and not the overall gene product. I'll talk more about this. And there's been this kind of historical way of thinking about discovery proteomics, where it's kind of like this triangle. There's the number of perspective and review papers that show a triangle where you start with a discovery in a small set of samples, and then you have a validation in a much larger set of samples. I'll argue that should actually almost be the opposite way around. That you want to have your discovery performed in a much larger set of samples, or at least a rectangular set of experiments. So we do need to have a very large sample size, especially if you're going to measure a large number of analytes. We have this massive multiple hypothesis testing problem, and we often kind of ignore this. People want a bigger list without realizing that often with that bigger list comes a requirement for a much larger set of samples. Okay, let's start for the first thing. So we already know about this. So the data-dependent acquisition's been kind of the workhorse methods. It was originally pointed out to assign, can we say presence of an individual peptide in a sample? You can't even say absence with it, but initially it was which peptides and which proteins were present. The main limitations for why we've struggled with it in our lab is that if you run the same sample multiple times, you don't necessarily sample the same things. and this limits it in many ways. The other thing is limiting is that the MSMS process, the MSMS spectrum, is 10 to 100 times more sensitive than the MS1 signal. So why are we using the MS1 signal in order to derive what we picked for doing the MSMS? That's the other limiting factor. So then the alternative to this is doing a very targeted assay where we measure a very few number of analytes. Our result is no longer a spectrum, it's a chromatogram, so that's kind of our measured signal. The ultimate goal is to extract information for a specific target peptide, which that assay was developed on and validated, hopefully, and to then ultimately become a quantitative measurement for that individual peptide. And in between, this compromise, as Brian mentioned it, is to do data independent acquisition, which was originally developed in about 2004 by John Venable. And the goal here was to keep this sort of systematic sampling so that you measured things in all conditions and all samples. But the downside, of course, is that we have some lack of specificity. Every spectrum is intentionally meant to be a chimeric spectrum. We are intentionally multiplexing the collection of our data, and that means that while we extract the chromatograms, we can often get clean chromatograms in order to be able to detect and quantify peptides. It does lose some specificity over target analysis. The advantage, of course, though, is that we can measure a larger number of analytes. I will point out that hardware is getting faster and better. It's not necessarily getting cheaper, but it's definitely getting faster and better. And once we hit the point of about 50 hertz, these two methods will become equal. So at 50 hertz, if you collect data from 4 massive charge wide isolation overlapping, like how Brian talked about earlier today, you get basically 2 mass to charge effective across a 500 to 900 mass charge range with a 2-second cycle time. So if you wanted to go to a 2.5-second cycle time, you could easily go from 400 to 900, which Brian showed basically covers most of what you'd actually want to be able to sample. So 50 hertz is that magic number, and you hear vendors all the time now saying that they can measure things at 100 hertz, right? So that should tell you, is there, at that point in time, I think this method basically goes away because you can sample the entire mass range systematically, reproducibly all the time with a isolation width that's very similar to what you'd get from a parallel reaction monitoring experiment.
And the resolution?
Pardon me?
The resolution?
The resolution? Well, I got Tim's talk, they say 100 hertz, right? And so that's top resolution. And there is, there are instruments being developed, we know, at Thermo Fisher, whether or not they'll make it to commercial, that are going at 60 to 70 hertz. They do sacrifice resolving power in the process. So I will point out, though, the precursor isolation actually is a little bit more important than I think the product time resolution. Okay, so one of the things that I've gotten pushback on when I've talked about this and saying that this may disappear is that some people said, look, if you were to actually collect your data, would you rather should you spend most of your time sampling areas where there's nothing there, or there's very little stuff there? Because that's what actually often happens in parallel reaction monitoring. You select the window at a specific mass, you're isolating that mass, and you're subjecting it to fragmentation. It's also a very similar thing to data independent acquisition, but because we're measuring multiple things at once, it's actually rare that we're measuring nothing in any one scan. But the argument is that you spend most of the time sampling the things that you know are there, where there's useful signal. So my argument to that is this argument also has come up in other fields. So in the field of astronomy, this was a huge debate with what to do with the Hubble telescope when it first went up into space, and the cost of doing the telescope time was astronomical, as you could imagine. So whether or not you should focus the Hubble on looking at these features in the cosmos that we know are there, but we want to get better information that are more high detailed resolution, or should we be monitoring and setting the telescope in areas of the space where we don't see anything, to see whether or not we can discover something new that hadn't been done before? So what they decided to do was a fairly simple experiment. they picked a region in the sky that's near the Big Dipper. It's a space that was the darkest region in the sky. And they focused it on an area that's about the size if you stood in the ground and held a pinhead up into the sky at your arm. That would be the region of the sky that they actually measured. Now they have the same sort of equivalent of automatic gain control. They basically said, okay, we're going to set and collect light in this telescope for a certain period of time, and they realized that, but given how dark that space was, that they're going to need to collect data for at least 10 days in order to be able to get a useful image out of that. Okay? Well, this was the image that they got out of it. In the end, in that pinhead of space, they found 3,000 different, completely undiscovered galaxies. Not new stars, not new planets. These are 3,000 new galaxies in the area that was in the pinhead previously. They learned more about what we learned about the universe in that single experiment than they had done in every experiment since before then. And since then, now this has been a majority of the telescope time has been spent doing experiments in this what they call these deep field experiments. Because this has been often the most profitable, is actually focusing on areas where there's little stuff there accumulating either light or photons or ions, in the case of mass spectrometry, for a long period of time to then be able to sample those things. Okay, so we need methods where so I think hopefully that makes an argument for why we like to do things by data, independent acquisition, and parallel reaction monitoring to be able to assign things very specifically and systematically, and to be able to go back to those things and make sure that we measure the same things over time. Okay, so we need methods where the change in signal reflects a change in quantity. So one thing that I'll point out is, are the measurements that you're making, are they quantitative or are they just differential? I'll point out that there's nothing wrong with a differential method. In most cases, most people just care, I want to find things that are different between one condition and another. But in other cases, it's really important to say whether or not there was a 2-fold change or an 8-fold change between two different samples. Right? If all you care about is between condition A and condition B, that there was a difference, then it's fine to have a differential method. So in order to do this, we actually need to define some things. We need to define things like limited quant and limited detection, which is non-trivial. So I'll point this out, right? And this is a using method that was done in collaboration with Olga's lab. That this actually is not easy. It's not straightforward. But in order to do this, all quantitative measurements have kind of this hockey stick type shape. In fact, in many cases, it has a sigmoidal type shape too as you go up higher. But in most cases, we're more worried about things at the lower level than we are at the upper level. So if you have these two measurements, the difference in intensity is in this linear range of the calibration curve. So we would often say that those measurements are probably quantitative. If you have a measurement down here where one of the points is below the limit of detection or limit of quantitation, and the other point is above the limit of detection, limit of quantitation, there will be a difference in the signal between them, but I wouldn't necessarily say it's quantitative because the change in signal is not a linear response from the change in quantity. Does that make sense? So all calibration curves, whether it's a BCA assay, whether or not it's a UV or fluorescence, etc., all have the same sort of limit. There is a point at which when you go down, the change in signal no longer reflects a change in quantity. We call it this limited quant. And there's a range at which you can no longer distinguish the signal that you're measuring from that of a blank or a background. So that's the two different levels. One is this limit of detection, and then when you go above the limit of detection, so you can actually detect the signal, we often define the limit of quantitation when there's a certain precision requirement that's often met. So we've decided to figure out ways in which we can do this in DIA experiments, really complicated matrices. Lindsay Pino and our lab, using methods that was developed in collaboration with Olga's lab for determining the limit of detection and limit of quantitation, decided to build on methods that based on this 2014 paper from Russ Grant and Annie Hufnagel, where they suggested taking a reference proteome and make dilutions in a similar complicated background proteome. So I'll give you a couple of examples of how we've thought about this. So in the case of cell lines, you can take an unlabeled form that's your reference, and you make dilutions with it with a 15N labeled form of that. And I'm not saying that we're using the ratio of them for quantitation. We're just trying to demonstrate that the signal is going down while keeping the matrix complexity the same. So all we want to do is dilute the signal in a linear form, and then be able to measure it. So obviously the matrix will stay the same. We can also digest tissue in our normal preparation. We can also take our background and digest it in H2O water, and then make dilutions with the digest that occurred in H2O water. We've also done it by making dilutions in an evolutionary diverged species. So actually, in this Grant and Hoofnagel perspective paper, they suggested for plasma using chicken plasma. We've actually used chicken plasma for human plasma. We've actually used chicken plasma for human CSF. We've also used chicken livers in some tissue, some liver homogeneous experiments. So you can actually use different species to make a dilution as well. Sometimes you can actually take different cell lines. So if you're interested in one specific protein where you have a protein present, you have another cell line that doesn't have that protein, you can make dilutions of that as well. Just to show you a little bit of what this suggests is we have here's a yeast protein, and these are different peptides that we've detected in the different measurement. This one peptide has an extremely low limit of quantitation. There are several when they're shown in the kind of peach colors where we actually can't quantify the peptide at all. The change in signal does not reflect the change in quantity. And the range over which these things are linear is different for each and different individual peptide. So it is very important that if you're trying to say that you actually do have a quantitative measurement, that you should actually say that the change in the signal is really reflective of the change in quantity. I'll point out that a lot of times people start to refer to this, especially in the TMT world, as sort of ratio compression. It's not really ratio compression. It just means that one of your signals is below the limit of quant and the other one is not. It's in that linear range. So it's as simple as that, is that they haven't defined the linear range of the measurement. So one of the measurements is below the limit of quant. It can get a little bit depressing, though, at times, because there's lots and lots of things that we can detect in certain samples and matrices, but not all of them are quantitative. So Brian did talk a little bit about this narrow window DIA measurement. So we're very interested in making measurements in cerebral spinal fluid. So if you look in cerebral spinal fluid in the narrow windows, there's about 8,600 peptides that we can find in that narrow window data that we can detect in it, which those things map into about 3,000 protein groups. When you go to try to transfer those over to the wide window data, there's 6,000 peptides in the wide window reference. I've actually asked Lindsay like 5 times a day, actually really exactly 6,000. Her experiments did show exactly 6,000, which reflect about 2,100 different protein groups. But if you then start to think about what fraction of those you can actually quantify, it's about half. So, which is about 1,300 different protein groups. So this is actually kind of a little bit disappointing. It's not saying that you can't use these ones to make differential measurements, but the ones where the change in signal will probably reflect the change in quantity is only going to be a smaller subset of the individual peptides. Okay, so I will point out that I actually wish that people started to report numbers like this as opposed to these larger numbers. We get to a point where everybody wants to have the biggest list imaginable, but this is the one that often ends up being the most conservative and the most useful. These are the ones that we can measure the most precisely and with the best accuracy, quantitative accuracy. It would be great at a point in time is that this became the benchmark for which we can then start to report our numbers under, as opposed to these sort of exaggerated sets of peptides that we can't do much with.
Do you have any idea what characteristics make something quantitative in terms of a peptide at this point?
Yeah, so it all comes down to whether or not that dilution curve, whether when you go lower down than the endogenous amount, do you actually get a decrease in the signal?
But like protein character, like the actual characteristics?
That's harder to predict. So one of the things Brian did at one point is he actually looked across different peptides of the same protein, and there was about 2 to 3 logs of differences in intensity for each individual protein. Some of it's just they're not released from digestion. Some of it's because they don't ionize as well. Some of them are just because they're imprecise. So some peptides actually kind of, if you sit them in the autosampler tube for too long, they'll kind of just disappear. That makes them imprecise. And if they're imprecise, then we say that they're not very quantitative. Yeah?
So what about just reporting the distribution in terms of the accuracy of quantitation? I like very much the distinction that you make between something simply being detected and something being quantified. But in reality, we always have some error bar that sometimes you can estimate quite accurately, other times less so. But what about just having a continuous distribution that reflects the error estimate?
Yeah, so the difference is, so let me try to straight. So you're saying precision. So reporting precision is what people have kind of defaulted to for some of these things. One point I'll make out is that you can actually have a precise measurement that is below your limit of quant, unfortunately. So that's the challenge here, is how do you ensure that it's above the limit of quant and in that linear range? But we basically, our limit of quant has to satisfy two criteria. It has to be above that limited detection, and it has to meet some minimum precision criteria. So we're taking that one extra step. So a lot of times people just arbitrarily set like a 20% CV threshold for things, and that is definitely a much better starting point than just going straight with just it's detectable from some search tool. So you can do it. So there are predictions. So most of the prediction algorithms were derived by DDA data, and so you can, and because those machine learning algorithms were trained on whether or not a peptide is detected by a DDA algorithm, so that means whether it was triggered on something, and whether or not that was then matched with a search algorithm. Those tools are very good at predicting whether or not you're going to sample a peptide and determine it from a search engine. So Brian has a tool that he calls Prego. It's actually an external tool in Skyline. You can actually install from the tool store. And that tool does allow you to, what he did that was different is he actually used independent acquisition data to train whether or not you'll find something in a targeted experiment. That tends to work much better. And one of the things that he tried to make a strong point in the manuscript was that he didn't feel that he had a better machine learning approach. He just felt like he had data that was more reflective of what people were trying to predict, which is that he had chromatographic peaks from product ions, which was more predictive of what most people are trying to do in a targeted experiment. So that's the closest thing that's out there. But now that we have much better and much more DIA data, it may be worthwhile retraining some of these things at some point in time. It's on the very large to-do list of things. Okay, so comparing signal intensities between labs and instrument platforms. This is obviously like an Achilles heel, right, for us, right? Data is collected in one lab and on one instrument platform, where you have two instruments in the same lab, and you're trying to make all the measurements on one instrument. The signal changes across time. You got the service engineer coming in, cleaning the instrument. The signal comes back up higher, and then how do you compare that with the signal that maybe a couple months later, and your quads are dirty, and you've lost half your signal in that period of time. So what we kind of make is this assumption, right, is that the peak area measurement should scale with the amount of peptide in the sample. So if you have some known quantity and some measured peak area, you make the assumption that a higher quantity should have a higher abundance signal. But it could be that you have a triple TOF and you have a Q exactive, and you want to be able to compare those signals between one another, and they have completely different arbitrary signal units, and so you may not necessarily be able to compare the signal from one instrument platform to another, or it could be a case where you have a lower quantity giving you a higher signal than that of a higher quantity. It's not entirely clear how all the vendors report their measures of quantities. Even if you go in Thermo Fisher, the Fusion platform versus the Q exactive platforms have very different measured signal intensities. The Quantiva and the Vantage from Thermo and the triple quads have a different measure of signal intensity than that of the Altus. So you really need to you can't compare these arbitrary y-axis signal intensities very easily. Okay, so the purpose of signal calibration is to place the signal on the same scale, measure the signal on the same scale. And we often think about this thing as absolute calibration. There's this term absolute. But the only thing that makes something absolute is that you're comparing it relative to something that somebody said was a certain quantity. And that certain amount of quantity was often done by another method that was often determined by some arbitrary series of reference values. In fact, we have an entire institute in our government system, the National Institute of Standards and Technologies, where their entire goal is to determine what are our references that we should be making our measurements relative to, whether or not it's the atomic clocks, whether or not there's certain amounts of things that we weigh a certain analyte relative to. There's an entire aspect of the U.S. federal government that's focused on this. In fact, if you were to look at the kilogram, up until this last year, all kilogram measurements were actually weighed relative to this piece of metal that was kept under armed guard just outside of Paris. And it was originally designed to be a piece of metal that has a mass of a liter, that approximates a liter of water at freezing point. It wasn't exactly that, because of course they were making a piece of metal, and so what the kilogram mass is actually this piece of metal. It was meant to approximate this, and it was there within a few kind of nanograms of weight, but it was not exactly the same. I'll also point out too is that every 50 years they had to take this thing out of storage. They had 6 other ones around the world, and they weighed them all relative to one another to make sure. And this one was actually losing mass over time. So even though it was being recalibrated, the reference material for which all kilogram measurements were based was actually losing mass. So if you weigh yourself in the kilograms, you're actually gaining mass over time. So you can kind of, it's in like the low nanograms over 50 years, but still, it was definitely differential. So the same thing's true with meter. A meter, it was meant to be 1/10 millionth the distance of the equator to the North Pole. But for other areas of science, this has been really arbitrary. So I came from an isotope ratio mass spectrometry background, and when you measured 13C, you always reported it as a delta PDB. And PDB was PD belemite, and it was a fossil that had been found in this carbonate rock formation in South Carolina. And nobody's been able to find that fossil again. So, and the reason why people measured it relative to that is because it had the highest 13C to 12C ratio that was ever found. So all the values were supposed to be less than that. So what NIST did is they made a reference material relative to PDB. People made their measurement, they calibrate their reference material relative to the NIST reference, which is relative to PDB. So you often would make your measurements relative to your reference, which is relative to NIST, which is relative to PDB. But you still always reported your values relative to PDB, even though PDB doesn't exist anymore. So that's kind of an arbitrary value, but in fact, we've been sort of talking about it within some of my clinical colleagues that maybe we should just have, so at the University of Washington, the clinical labs, almost all the ELISAs are measured relative to Andy Hoofnagel's plasma. So he's a clinical chemist. He drew his blood, and they used that as the reference material. So we just said, well, maybe we should just have Hoofnagel units or something like that. And it's kind of got this joke going around. But it's you can kind of get the sense, right? Because that's basically what it is. What is the thing? As long as everybody makes their measurements relative to the same thing, and that same thing is not changing, of course, if this is changing, right? But then it's a fairly constant thing to make our measurements relative to. For standard mean ocean water, so if you're making 18O measurements, there was a guy named Harold Craig. He went around all the oceans. He filled up a bucket with ocean water. And he said, everybody's going to make their 18O measurements relative to my bucket. Well, that bucket's empty now. And now we're making measurements relative to other standards that were measured relative to his bucket. But I think you get the point, right? Is that absolute quantitation doesn't really exist. The goal here is to have a reference value of which you call it a number, that the community eventually agrees on that number, and that becomes a reference standard. In fact, there's now been some discussions that maybe NIST will host things like plasma values, and then what we'll do is eventually crowdsource the actual abundance of proteins within that plasma sample, that then we get to enough consensus of what we call the amount of that protein in that plasma sample. If not, everybody just assumes it's 1, right? Or it's a delta value from that individual value. But it's nice to have an actual assigned number, right, to it. Okay. Pardon me? Yeah. So now it's actually done by physicochemical measurements. Like same thing with a meter. Meter used to be a metal bar that was found. Now it's like the time. It's the distance that light travels in a certain period of time in a vacuum, right? And so these things are much more. It's easier to reproduce more regularly. And part of this issue of this being irreproducible generated that desire to change the definition of kilogram. As I said, up until about a year ago. It was definitely this piece of metal. Okay. All right. So our goal is to take different uncalibrated values and put them on the same scale, right? Even if that scale is somewhat arbitrary. We like to have it less than arbitrary, but it's still somewhat arbitrary. Okay. Our lab, we love this paper, this lab check paper from the Aebersold lab. And our lab does a lot of experiments in yeast. This paper came out in 2015. And so one of the goals was, this data was all collected on SCIEX mass spectrometers. We do a lot of similar yeast experiments. Can we get the same values that they reported in Switzerland, right, on SCIEX mass spectrometers in our lab? Prepped from different batches of samples on Thermo Fisher or Orbitrap mass spectrometers. It's kind of a bold statement, but that's what we really want to be able to do, right? So what they did is we noticed that they had a reference. It's a reference that we often use. It's a BY4741. So BY4741 is the yeast strain that was derived from S288C, which is the strain that the yeast genome was done under. Sina Gamma-Magami in 2003 had this paper when he worked for Aaron O'Shea and Jonathan Weissman. He actually determined the quantity, gave a copies per cell for 4,000 of the proteins that were in that BY4741 strain grown under mid-log phase. They also had another experimental condition where they did osmotic shock. So basically what our goal was is, could we use this as our calibrant, as a common reference? Could we grow up our BY4741 strain? And could we calibrate our osmotic shock quantities relative to this and put it on a copies per cell, because this values of each protein that's in there has a quantity that Sina Gamma-Magami has derived it to be as a copies per cell. Does that make sense? Okay. All right. So the idea here is that you have a bunch of reference materials, you have a bunch of experimental samples. You may have samples done in different batches. You may have them done on different instrument platforms, either on a triple quad or on a QTOF, triple TOF. You could have it potentially DIA done on different days. So you can make measurements between days, between instrument platforms, and you can also test it between laboratories. So the first thing that we did is we took their data, and in there they actually used they had SRM data to validate some of their SWAT data. And they actually said, the triple quad data doesn't actually give the same signal as that of their QTOF. And you can see their SRM peak carries are fairly correlated, but they're actually, not very well calibrated with one another. If we express them all relative to their reference that was measured and prepped and measured within their same batch, and actually has a value associated with it as in copies per cell, their actual data is extremely well calibrated. So in their paper, they made a case saying that they can't compare their SRM data directly to their SWAT data because it's poorly calibrated. But one thing that I'm trying to point out is that their data, when you use their own reference material in their sample, is extremely well calibrated and can be compared between the different instrument platforms. So Lindsay in our lab, as a graduate student, she actually did the same sample prep between 2 different days and measured on the same instrument platform. And it turns out that if you had the same person prepping 2 different batches of samples and measuring on the same instrument within 2 days of one another, they're still fairly well correlated, but it doesn't hurt actually to calibrate them and put them on a common scale. Right? So then if you take her data and you measure the same peptides by SRM that the Aebersold lab did, measure them compared to our QTOF data, they're fairly well correlated, but they're not ideally correlate, but then when you actually calibrate them relative to the common reference, it's actually much better. So we're using a single point calibration relative to a common reference material, where we've said that copies per cell of the protein are there based on the Gamma Megami paper from the O'Shea and Weissman. And then we went the last little step, and that is if you take the QTOF the QTOF data from the Aebersold lab and you take our quadruple Orbitrap data, the Q-Exactive data, they're really not correlated or calibrated very well. But when you calibrate them to a common reference material, they actually are very easy to compare between the two different labs. This was collected again in this data was collected it was reported in 2015. This was collected in 2017, reported in 2018. You know, this is 3 years apart from one another, different countries, prepped by different people, measured on different instrument platforms, but yet at the same time you can get the same results between different labs if you use a common reference. Now, I'm assuming that some of these reasons these outliers are here are because our common reference is not the exact common reference between their lab and our lab. They are the same strain, but they're not the same culture of cells. So that would be something that could be definitely improved upon. Okay. So basically, in all cases, the perpendicular offsets from the data points are improved dramatically when you're using a single point calibrator versus that from which you're using uncalibrated data. Yeah.
So I kind of missed it. So how exactly would you now calibrate according to the values reported in this paper? So
Yeah. So you could use BY4741 strain as one of your references too. That's basically what we've done between us and them. Or we will happily send you a culture of BY4741s that you would prep with your each batch, and that you would measure relative to your samples.
So they kind of had the amount of cells reported?
No, it's just done by the same amount of from a BCA assay, the same amount of protein from one versus another. So it's just a lysate. So it's a lysate. In fact, I think we actually started from the same cell pellet, but we may homogenize the cell pellet together with another pellet. We measured the density from using OD. We had matched the number of OD. We homogenized them together in the same batch. We then digest them together. So that's the key thing. It's not something that can be a digested sample. It's got to be something that goes through your sample preparation with you, because there is a lot of variation that can occur from one batch to another, and one lot of trypsin versus another lot of trypsin. So it is something that's got to get prepped with your samples at the same time.
Excuse me, if you have human biopsy samples,
Yeah.
how would you deal with reference with the-
Yeah. So we've done.
Do you think that we should do a data bank of reference system for different cancer cells and primary cells?
So one of the first things that our lab often discuss when we take on a new project is what will that common reference be? And let's start allocating enough of that reference away so that when someone comes back to us in 5 years' time, we can make measurements relative to that same reference. So in a lot of just tissue samples, it tends to be a homogenate from an often mixture of many different animals or different tissue samples. But in a case of some biopsies, it may need to require a pool of many different tissue biopsies.
Same with the biological fluids. Would you do a
Biological
pool?
fluids are fairly easy, so actually we've kind of urged, my colleague Andy Nagel has urged Golden West Biologics to make, there is a pool of 400 healthy individuals for plasma. That is a very great common reference. They do sell it lot to lot, so you'd want to make sure that you have the same lot each time. We've used their CSF before too. It's not as applicable because it's from a much smaller number of patients. So in our case, they tend to have their CSF from a healthy pool of middle-aged people, when a lot of the CSF that we're looking at or interested in is from elderly populations with Alzheimer's disease, or Parkinson's disease. So it's a very huge difference in age effect. So it's quite a bit different from it. So we've made our own common reference from CSF samples that we've collected over time through either our hospital or Stanford's hospital. Yeah.
So, looking at that first level plot, the one with of your 4 plots over there, so I think I can explain why the shape changes, but then could
Yeah, I'm assuming, so right here. So lower signals have worse relative precision in general. But when you calibrate things to copies per cell, the lower signals may not necessarily be the lower copies per cell. That's my explanation for it. So you've moved some of your worst precise things up, and you've moved some of your better precise things down.
Yeah, so there may be things with lots of copies per cell that just don't have it.
That's right. That's right.
So and then if you go down to that one, so I was trying to figure out that one because there's this weird. It doesn't look like just variance. There's kind of this weird.
No, that's I'm almost certain is an artifact that we're not using the same reference, right? So Natalie grew up BY4741 strain from their clonal isolate, right? They harvested at mid-log phase, and then they used that in their preps. Lindsay grew up, another different set of BY4741, probably collected decades later or years later, and grew up, and so there's differences in media, etc. So this is a worst case scenario. If we actually use the same set of cell pellet from the two, it should be orders of magnitude better. This is a worst case scenario, and it's still working hugely.
I actually kind of wondered whether some of it may also be the compression of the slots. It's like that it goes down to one and kind of gets sloshed there. So
Yeah, maybe.
that makes some sense why you would have a spray off
Maybe.
to the side.
Yeah.
Because it's saying basically that there are things that are expressed as being lower in swath than they are in the IM. So I think
Okay.
some of it,
maybe that's possible
some of it
too.
may be the fact that the swath signal is kind of like, it's not a full distribution. It's getting squished down to one.
That's possible. That's definitely
So
possible.
that might
I was just assuming that we were shocked actually that it worked as well as it did, and we were kind of, and our assumption was just we're not using the exact same reference. That became an argument for us why we should be using the exact same reference. Yep.
So basically you're saying that if, for example, you and the lab in Zurich happen to have the same spike and you wouldn't need anything with known absolute amount at all.
Right.
You would just, without knowing the amount, as long as it's the same spike in.
That's right. You could measure it as in, the MacCoss lab reference units, right, as relative to 1. It won't fit a line there. The reason why this fits a line now is because we've given each value for the proteins as a copy per cell based on the Gamma Megami paper. If it's just all relative to 1, it'll be just one circle. Yeah.
So I do want to make a point, though, because you said spike in.
Yeah, it's a spike in. Yeah, that's right.
It's not.
Yeah, that's right.
Spiking in anything. In fact, that wouldn't work.
That's right. This is an external single point reference calibrator.
Basically, if you had a spike in, this probably would be better, right?
No, it has to be external. This is external calibrant. We're calibrating this thing to an external single point calibrator. It would maybe be better if you had a multi-point calibrator. So we could make dilutions of this and then use that, but we're using a single point calibrator.
But the only things we can quantify across labs are things that are found in
That's
the
right.
sample.
That's correct.
So it needs to be the full proteome in the full proteomes matrix.
Yep. Yeah. So I'll point out, this is exactly how when you go and get blood drawn and you get measures of proteins measured, it's how the ELISAs are calibrated. They are calibrated relative to a common pool of plasma sample. There is no spike in for an ELISA, it's just a signal. They have a single point external calibrant that they calibrate that protein level to, and everything's measured relative to that, and then that common reference is given a number either by some calibration values from the manufacturer or from NIST or et cetera. So that's how it's done. So we're using an external single point calibrator. And I'll also point out too that LabCorp, as for several of their clinical protein assays, has derived that using a common external single point reference is a way better calibrator than using internal stabilized labeled peptides, because the sample matrix has to go through the same sample prep every single day, every single time. And if the digest only goes to like 90% one day and 75% to another day, that's accounted for by that same sample prep.
I think you answered that question. Basically, a single point calibrator, external calibrator, the way it was acquired is the same way we will process and acquire our data, the same exact.
Yeah, you prep it every day. So every day you get a batch of 10 samples or 20 samples come in, you take one of the reference samples out and you prep it with it, and then you measure the 11 samples. We often actually have 2 references in our lab, and one is we calibrate all the samples with, and the extra reference we use as a QC, and we know that the changes relative to that reference are always going to be the same. So we don't see the same changes relative to that reference in that QC sample every day we throw out the batch. We say there's something wrong.
Sorry, you use that reference for sample prep and also for the instrument itself?
Yep. Yeah, so there's 11 measurements now. So there was 10 samples, you have 1 reference, all the values are expressed as a relative measurement relative to that common external reference. It's like having an 11th bridge channel, people call it for TMT, but because we're doing it all as separate runs, there is just an extra 11th run.
So it may be a trivial question, but your final units are running molecules per cell as a log function. So of course that you cannot plot log area in the end. So do you have an interconversion factor?
Well, in this case, because of this Sina Gamma Megami paper, in that cell line in 2003, they provided a number for the copies per cell for 4,000 of proteins.
Is this a number which you can apply to any type of
For that cell line. For that cell line in mid-log phase.
so then you need to optimize for everything.
No, so if you didn't have a value, it'd be a percentage relative to 1. So when I mentioned like PDB, like PD belemite, we used to call it as, it was all per mil, so parts per thousand, because our precision was so good, everything was in parts per thousand. But in the case of here, we'd probably just report it as a percent relative to our reference. In fact, we even mentioned this in the paper, saying if you didn't actually have a number associated with it, you just report everything as a percent relative to the reference. And we talk about other fields where they do the same thing. They're good questions, great questions. So also I'll point out too is that this is something that's kind of a pet peeve of people who kind of go through my lab. They kind of get this, is that a lot of people try to report values on the protein level, right? So every protein has a number, and they try to report the values, the outcome of the values as a protein measurement. So every time we try to do that, we've often missed the most important things that are often known to be involved in the sample. And I'll try to explain why that is, and I'll try to explain why I think this is a problem, and why I think it's actually hard with a bottom-up proteomics experiment to actually report a protein level. Now don't get me wrong, that's what people want to do, but what we're measuring is a peptide. And we know there are a lot of proteoforms. In fact, if you ever hear Neil Kelleher talk, he'll go on and on and on and on about the importance. He wants to measure 1 billion proteoforms, but actually it's really important. There really are proteoforms, and they're complicated, and there's a lot of proteoforms that are still completely unknown, which we know that some proteoforms are disease specific and some are not, and it's really hard to sit there and try to wrap them all up to the protein and what even is the protein. And I'll give you an example here in a bit. Right, the functional form of the protein is not known in many cases. Okay. So if I just took one protein and drew it as a squiggly line, it doesn't really exist in that context. It's more something like this, right? You have multiple different phosphorylations. You have some on the same molecule. You can have other cases where you have a single phosphorylation molecule. You can have truncations on both sides. And this is a very simplistic case, right? There's been some cases where people have run 2D gels, and something like 30, 50, or 100 spots have all been to the same exact gene product, right? So these can be very complicated, right? And they're all, of course, at different levels. So if you're making a measurement on the peptide level, it's going to be the weighted average of the different forms that different peptide is present in. And of course, they all would be potentially moving in different directions, and they should be moving in different directions. In fact, there's enormous cases when you start, especially when you look at human proteins, that the peptides are moving in different directions across different conditions, right? We see this time and time again for different experiments. Every time we look deeply at it, we tend to see that. So I wanted to kind of bring up a couple of experiments that have been going on in the lab in the last couple of weeks, and these are just kind of highlight some of this. So if you were, the only 2 biomarkers that we have for Alzheimer's disease is A-beta and tau. They're the only ones they're the 2 most common ones, at least. There are other ones that are kind of being in development. So A beta is derived from a peptide. It's from the transmembrane domain, from the amyloid precursor protein. Most of the A beta peptides are not changing, with the exception of these ones down at this end, between different conditions. This is from about 75 different samples. And I'll also, let me just describe a little bit of what these four different groups are. So there are those that have genetic Alzheimer's. So these are people who have a known mutation either in the amyloid precursor protein themselves, this one, or the protein that's responsible for processing the A beta. So this is the presenilin. Then there's another group, which is those that have Alzheimer's dementia. So these people have dementia, and their brains looked like they had, that a pathologist said that they had Alzheimer's disease. Then there are those that are derived as being that are clean. So these have no. The pathologist says they have no clear pathologies that link them to having Alzheimer's disease, and they didn't have dementia when they were alive. And then there are those their brains looked like they have some sort of the pathology of Alzheimer's disease, but they were cognitively fine. They didn't have dementia. So when you start to look across the different levels, there's two things that are fairly striking. One is that the amount of these two peptides in the A beta peptide for this with genetic and the head dementias are through the roof relative to the other peptides. If you actually look at those that have the Alzheimer's disease dementia, this level relative to those that have no observed pathology in their brain, there's still an enormous difference. It's about a 10-fold difference still between that of the clean brain and that of those with the Alzheimer's dementia. And then those with 30 resilient, those that have no dementia, but their brain still looks like they have some of the pathologies. It has kind of an intermediate level. Now these two peptides are changing dramatically between the different conditions. Most of the other peptides, with the exception of this one, are not changing nearly as much. If you were to look at the statistics across the entire protein, there's actually no real statistical difference if you merge them into a protein level quant. Now, this has been known for almost 30 years as being a reasonable biomarker and diagnostic for Alzheimer's disease. And some of these changes are actually on 40x fold, right? So these are actually pretty significant changes in the mass spectrometer, and yet they'd still be ignored in many cases. Here's the other main biomarker, so tau. So tau is another protein that's involved in it, and it also has a significant number of proteoforms. The same groupings here. So those with the genetic Alzheimer's have a really high level of tau, and those with Alzheimer's disease, the same regions, but only in this level where it's this microtubule binding region of the individual protein. There's also a change that's occurring here at this 195 peptide, 194 and 195 peptide, where the peptide here in this Alzheimer's dementia goes down a little bit. This is a peptide that spans the serine 195 that's known to be hyperphosphorylated. So the stoichiometry of the unmodified peptides going down a little. It's still not significantly different in the sheer numbers that we have in this case, but it still is an interesting observation. So one thing I just want to remind people is that what we're measuring are peptides. There are proteoforms that are different between the different conditions, and the peptides are spanning different forms of proteoforms. I want to kind of bring up another couple. Here's another one associated with tau. There's two different alternative splice forms of tau, those that are in fetuses and those that are in adults. And we were given samples this was done by Roche. They sent us samples from cell lines, and we measured the peptide that was spanning this alternative splice form from this fetal tau version. And we were easily able to see that it's different between the groups of sample 1 and sample 2, but with peptides that span in this alternative splice form, there's only one peptide for which you could derive that alternative form, right? So again, this is something that you can only do on the peptide level, and that's what we're actually measuring in most cases. And there's another case, too. We were looking at amyloidosis, Hanyan Yang in our lab. Not only we were trying to type whether or not an individual sample had transthyretin in it, but she was also able to work out that she was able to find this H to R peptide from two different peptide forms. Here's the peptide here, the shorter peptide, and there's also the peptide here, the slightly longer peptide. So she was able to derive that not only was transthyretin the main protein that was in this piece of amyloid plaque in this tissue that she was given, but she also was able to derive that the patient was heterozygous for a polymorphism that spanned increases the risk factor for amyloidosis formation. The final one I want to give it as an example here, and this is one that our lab struggled with for a long time. And this is Andrew Sturgachis was trying to do an experiment looking at chromatin. I won't go into too much detail, but the main thing was he was trying to take chromatin and fractionate it. So those that are loosely bound to chromatin and those that are tightly bound to chromatin. We measured this one transcription factor, Cap1, and he made an assay to 3 different peptides, and he looked across all the different fractions, and everything looked fine with the exception of this one fraction, where one peptide was hugely significantly different from the other ones, from the other peptides. And he repeated this experiment about a dozen times, knowing Andrew, that's the way he often does it, and he still couldn't figure out why this one was so much lower than the other ones. Partially through this project, ENCODE released all their RNA-seq data, and in this K562 cell line, they noticed there was another third alternative splice form. Now notice this peptide's not present in that one. And while it is present, it isn't present in this HepG2 cell line. And we went back and we actually collected Western blot data, but of course whenever we do Western blots we have these thin little cutouts because we often expect lack of specificity. And we went back to Western blot data which was an antibody to 7-dome, and there actually was an extra band that was down there for recognizing that protein form. So the mass spectrometry data is right. You should not expect peptides to change in the same direction across different samples. There are different proteoforms. Often, whenever we've dug in deeply to try to understand why different peptides are disagreeing with one another, it's actually always been interesting. We actually there are a couple people in the lab trying to look at whether or not they can look at co-expression of individual peptides to come up with peptide grouping within individual protein groups. But that's still kind of work in progress. So the final thing I want to kind of hint at before I go on to another topic is it's mostly kind of an argument, and that is we need to have start having experiment. Yeah.
One question to this: what if we see these effects for different peptides, but we cannot figure out what's going on, and still would like to come up with a value for protein? What would you suggest?
What's the protein? So okay, so this is known here. So which value would you give it to? Right? It would be kind of it's hard, right? These are not straightforward to give. We know that there's no protein. We know that protein exists as different proteoforms. We just don't know what those proteoforms are. So I'm not saying that we don't want to come up with. So biologists often ask us, give us a list for the protein, but whenever a biologist studies a protein deeply, they know that we've got 3 different proteoforms. There's 4 alternative splice forms. People know. We know that this one alternative splice form is only present in cancer and it's not present in another case. And so people who study, the biochemists who study the proteins or cell biologists study the proteins really deeply, they know that these things are an issue, and they'll often come to you and say, your data doesn't make any sense because we know that there's this proteoform there and your data is not reflecting that. In fact, one of the earliest cases we did, we were working with, one of the first targeted assays we made was to a protein called surfactant protein B. And it's like this 500 amino acid gene that gives rise to a functional protein of 79 amino acids. The postdoc that built the assay to start with just made peptides to measure the whole 539. And then we made these measurements, and the peptides that were coming back the most were actually ones that were on the carboxy terminus. And the scientists that we gave the data back to said, it can't be right. You're measuring these peptides in the carboxy terminus, it's not the functional form. It turns out our data was right. And the reason why these babies were suffering from respiratory distress is because that portion of the protein wasn't being cleaved off correctly. And their antibodies weren't recognizing that form of the protein. So in general, I trust the data that we collect. And the problem is right, we just can't interpret it yet. But it's really kind of exciting whenever you kind of go back and try to figure it out. Okay. So I'll try to make this case. So if you look up almost every review or perspective paper on biomarkers, they'll make a case that there should be this discovery phase, which is done on small numbers of sample sets or maybe pools. And then there'll be a validation where there'll be a larger number of samples that are done for a validation. And in many ways, I think this is slightly backwards. Because of multiple hypothesis testing, it's really hard to find differences when you're measuring large numbers of things. And actually, you can get by with sometimes validating things with because your list is now a much smaller set that you're validating, a smaller number of hypotheses that you're making on a smaller group of samples. So often it may end up being down the road that we actually do our experiments that are a little bit more like this. But I do want to make sure that people are aware of this, because it's, if nothing else, just a hypothetical experiment, that it's often good to work before you start doing your experiments, to work with a statistician who might be helping you with these data sets to ensure that your data is correctly powered. One of the things that often we get told to is like people start to cringe, the statisticians we often work with, including Olga at times, whenever you start to say, you're going to be measuring 30,000 peptides, they're like, oh my gosh, you're measuring 30,000 peptides? What are we going to do with the multiple hypothesis testing? You don't have enough replicates in any of your number of conditions. And I just want to make sure that everybody's very clear, because everybody says, well, I can measure more things by DDA in a TMT experiment, but I can only measure 10 samples, right? But the key thing is if you're going to be measuring 30,000 or 40,000 things, you need to be thinking about hundreds, if not thousands, of samples often in order to gain things correctly powered. So here, just again, these are from a fairly old review. It's in a completely different field, but again, these plots can be done easily for proteomics. But it just basically shows the difference in power. So if you had a 99% power at a single sample, so a single analyte that's being measured, and now you're starting to measure something like 10,000 analytes, you're now going to have less than 50% power, right? It's basically what the figure is trying to show. And a similar sort of thing's being shown here. You can also look at the effect size. So if you originally had a certain effect size of 3, now you're measuring 10,000 analytes, you're now going to need to measure 3 times 1.5, right? In order to be if you measure a 3-fold change, now it's a 4.5-fold change. Basically, you need in order to see that same power, the same significance. You could also look at it as in the context of the number of samples. If you needed 5 samples to start with, and now you're going to be measuring 10,000 peptides, you need to basically multiply your number of replicates by 3, right, in order to gain the same power. I just think that's pretty important, because most people don't think about that. When they do experiment, people want to measure more analytes, but they don't realize that what you're doing is making it harder to measure the analytes that would have been able to be measured easily in a smaller sample set. So it's that trade-off. You got to be careful for what you wish for, right? People want to be able to have methods that measure more analytes, and I definitely feel like we need to be measuring analytes at lower abundance and more specific to certain pathways, either transcription factors or kinases or other regulatory enzymes that people are interested in. But I'm not sure that we should be making very large lists of imprecisely measured data, right? So again, there's this list of how many things can you detect versus how many things you can quantify, and then ultimately there's how many things are you quantifying and how that's going to affect your power. Okay. All right. So I just want to make that kind of a little bit of a push, because I actually saw something got written up on one of the boards that how smaller sample sizes are sometimes better. So I want to kind of pitch my viewpoint on that as well. So I do want to kind of give in the last 30 minutes some kind of limitations and complications of data independent acquisition. So one is wide isolation windows results in loss of selectivity. Results can often be incorrectly exaggerated. So I think when you go and look at some of the data that's in the literature, you may find that with some of the newer technologies that we have to start analyzing data, that some of the original publications may actually have had exaggerated numbers. I'll talk about how some of those things can occur. Narrow windows offer significant selectivity enhancements. We talked about the 50 hertz is kind of like that magic number. There's kind of super resolution-like methods to improve precursor selectivity. Brian already talked about this a little bit, which is the overlapping windows, but we can kind of talk a little bit more about that. Positional isomers are a problem, and DIA actually offers some unique capabilities to address them. So let's just start with this, is that DIA is always kind of a balance and sacrifice. Brian kind of already discussed a lot of this. You can kind of alter there's like 3 things that get affected. There's basically, for a given instrument speed, you affect the cycle time, you affect the mass range, or you affect the isolation width. Or you affect the throughput. So you can make multiple injections with narrow isolation windows. Narrow isolation windows is better. So with the narrower the isolation windows, the more you can detect individual peptides. So the detection of peptides in DIA data is inversely proportional to isolation width. So if you had a 2 mass charge isolation width, you'd get more than double, 2.5 times that of which 10 mass charge isolation width. That's huge. That's a big, significant difference, right? And so, so you do have to kind of weigh the difference of this throughput versus this sensitivity. So to overcome some of these things, has been the methodology our lab's kind of based on, is we use these 6 gas phase fractionated samples. It's like the specific method that the Goodlet lab developed. And we use either a tool like either a spectrum library tool or like a tool like Pecan or Walnut. This gives us basically PRM quality data. So basically, if you can't find it in a 2 mass charge wide window, you're never going to find it in a 12 mass charge wide window. It's the way we think about it, because of the data that we've sort of shown here, right? So we start off with this pooled sample. This defines what things are in there. These are the things that are detectable within our pool. We can then use the retention time and other pieces of information that were derived from these on-column detection measurements to then sacrifice the precursor resolution a little bit to be able to map these signals over to the 12 mass charge data. There is problems though. And this was an observation that Brian made kind of fairly early on, and I think Brendan had also made similar sort of observations too, and that is if you start to look at the data from the 2 mass charge isolation data, and you start to map the data from the 2 mass charge isolation data over to the data from the 24 mass charge wide isolation data, we started to make an observation, and that is that the best scoring thing didn't necessarily have the same retention time. Notice there's this group of things that are right here. In fact, they almost always had a slightly, I think it was, they always had a slightly earlier, earlier retention time, or later retention time in the wider window data, which just couldn't happen because all this blue data for doing the alignment is where 99% of the peptides are all falling. So why is it that these things are all mapping? It turned out all of these peptides had a methionine. And almost all of it was where an oxidized methionine peptide was mapping to an unmodified sequence. And I'll show you an example of this. This is, again, a figure that Brian made. And so what he basically did is said, okay, if I look for this oxidized peptide in a 2 mass charge wide isolation DIA data, this signal at 21.5 minutes is the one that scores the highest. If I do a 2 mass charge wide isolation window and look for the data that actually is not oxidized, this is the one that actually shows up at 27.5 minutes. So it's a huge retention time difference. If he looks for this oxidized peptide in a 24 mass charge wide isolation window, the thing that scores the best is actually this signal down here at the 27 minute retention time. Now the reason why this is happening is that both the unoxidized and the oxidized form are now falling in the same isolation window, and they share the entire Y-ion series. And this signal is about more than 10 times more intense than the oxidized peptide. So what's happening is that even though there is a difference in the 2 amino acids at the end, the 2 B ions, those B ions are underrepresented in ACD. And so this one scores higher, this signal scores higher at the incorrectly modified form than it should have. So what you've basically done is we've said now this oxidized peptide is present, but we've mapped it to the unoxidized signal. This is going to be a common problem if you don't have really nice and tight retention time information. Now, given the fact that we have a chromatogram library that says we know that the oxidized peptide shows up in this narrow retention time window here, we can exclude this based on using retention time. But without having that information, it would be really hard. So if you had only the ability to predict retention time within plus or minus 5 minutes or so if you want another, it would actually be quite a bit harder. So one way also to solve some of these problems is to... Yeah.
It's very important. I mean, so we don't have any kind of way to know from DIA exactly the ions capturing the oxidized amino acid. There is no criteria, like, I don't know, you said something about the intensity, so it's not like when you have an SMS and you have NC mass ratio charge, so there will be constantly a problem. So there are other PDMs here.
Yes. So simple answer is yes. This is a problem. And the only real solution here is actually to localize the modification specifically to the residue. So you could basically say, look, we're only going to accept an oxidized methionine if we have a fragment that says specifically that methionine is shifted by 16. The downside here is that would be a B2 ion, which would be a fairly prevalent and not be very specific in this case, and a lot of B ions are kind of underrepresented. The other thing that's a problem is that a lot of modifications may not be known. So what if you have a sequence variant? Let's say you have a serine to threonine sequence variant that may not even be known in that individual. You could have the serine, the reference form, incorrectly mapping to a different mutation that's only shifting the mass by 14.
So can't you use then the DDA spectral library exactly one under the same condition always? If there was a reference.
You need the retention time to be really tight. Good point. Which is why the chromatogram library offers us a big strength here.
So one figure that Mike doesn't show here, though, is that those two spectra for those two peptides, if you collect the DDA spectrum for them, they look identical because the oxidation doesn't really change the aggregation very much. So
In this
realistically,
case,
Yeah.
but in other cases, the ions associated with the ppm are quite different.
sure. In many cases, the ppm, so phosphorylation, for example, the ppm mass is so much larger that you don't have to worry about it. You're never going to get a mass shift of 80 unless you're looking at variable with window data, which is, again, why I stress variable with window data has some very big caveats. So that's definitely not an issue, I think, for these narrower
So I think the way we're trying to advocate here is that if you start with collecting narrow window DIA data with 2 mass charge wide window, but the only thing that will really get through is deamidation and citronylation, right? Are the 2 modifications that will get through. Citronylation, though, changes the cleavage site, so that actually is something that can be derived differently too. So, but the deamidation is the main thing that can get through. And there now, because these on-column libraries have this retention time measured very specifically on that column in that sample matrix, the retention time is known very well. So that gives us a lot of extra power. And something like oxidation does cause a big shift. You just have to be careful going with a spectrum library directly to a wide window piece of data. The other thing I'll try to make a case of is that our lab has tried for about a decade now to do DIA where we have less than 4 mass to charge windows. And the main reason for 4 mass to charge is that it's the size for which oxidation is always in a different window, and also CH2. So most sequence variants are caused by differences in CH2, so valine to leucine, serine to threonine, aspartic acid to glutamic acid. And so with a 4 mass charge wide window, doubly charged is 7, and triply charged it's 4 and a bit. So it really are those 2 things. You need to have your DIA windows less than 4, and you need to have your retention times to be fairly accurate. Yeah.
How good do you think is the retention time dimension? If in this case you have anyways different precursors, but if it's just a swap in amino acids, does it often?
Yes, it does. Even deamidation causes a fairly significant shift in retention time.
So in almost all cases, it really.
Well, I'll show you cases in a bit for like positional isomers where it doesn't always. And there you really can't do much unless you actually do have fragment ions that distinguish between the different positional isomers. Okay.
So is the ion mobility?
Ion mobility could be an extra piece of information. In most cases, though, the ion mobility is kind of used like a precursor mass. So you have ion mobility, CID, time of flight. And in that case, it's hard to predict ion mobility, right? So it's easier to predict mass. So ion mobility works in many ways like a precursor isolation. So, but yes, if you were doing like a timsTOF where you had ion mobility and you had a quad isolation, then you gain an extra piece of information where it could be in a different mobility, the oxidized form. All right, so I won't go into this detail, but this is basically, Brian already mentioned this. We feel like this is a really powerful strategy. It gives you a tremendous power to basically, it's like the equivalent of making your instrument twice as fast without having to get an instrument that's twice as fast, right? And I'll also point out too, it's fairly easy to do. And there was a lot of effort put into making this work into MS Convert. And we have, like we have on a lot of the websites, thanks to Nat and other people, we have, a lot of these tutorials that are put up there. So we have a tutorial on the Skyline website that tells you exactly how to demultiplex these overlapping window data using MS Convert and generate MZMLs. So I do want to give a couple, an example here, and that is, positional isomers. This is something that Brian's worked on a lot, and so consider these two peptides. The mass is exactly the same, right? They have the exact same elemental composition. There's no amount of mass accuracy is going to gain you ability to distinguish between those two. There may be a couple fragment ions that differentiate between them. The retention times may or may not be different. Now, of course, the site of these these two phosphorylations actually change the function quite dramatically. So this is a figure I kind of like as well. Is if you take data, plot it in this this 2 dimensions, and you kind of blow up this region where it looks like there's not much data there. There's actually a lot of data there still. Like we have this incredible dynamic range, but almost all of these peaks here have multiple chromatographic peaks. So this one has basically 3 peaks at the exact same mass. These are the isodistributions going in this direction. There's 2 chromatographic peaks right there. This one right here has 2 phospholipeptides as well. You can imagine this being a nightmare to do by data-dependent acquisition. You would trigger on the first one and exclude the later one, right? So this is a problem that really data-dependent acquisition really struggles with. But the only real benefit we gain here is that we actually collect data across time. And the nice thing is if you make enough injections of the same sample, as one of the things that Brian mentioned is that you don't get much overlap in sampling the same phospholipeptides. So you eventually start to see all of the different positional isomer forms, but you don't necessarily get it in any one sample. So from a paper that Brian was a part of from Judith Vien's lab, over 50% of the singly phosphorylated peptides have another positional isomer. This is pretty scary when you think about it. We've kind of thought about being able to link an individual phospholipeptide, but basically now we're basically saying, and this result from this paper in 2016, that greater than 50% of the things that have have a single peptides that have a single phosphorylation site have another positional isomer. So here's just a couple of examples, and this is in bioRxiv, and we hope it'll be out pretty soon. Here's 2 different positional isomers, localizations just swapped between these 2 different serines. There are 2 different. You extract all the product ions. There are 2 different chromatographic peaks. If you start to then just extract the positional specific fragment ions, you can begin to see 2 different forms over there. So this is an advantage of data-independent acquisition or parallel reaction monitoring. In fact, Brian started to see these things earliest by doing parallel reaction monitoring. When he started to measure a specific analyte, you notice a lot of the peaks actually had multiple retention times. Yeah.
I often see it in these figures. You have this small bump.
Yep. That's right there. So have you ever read any Gavin Reed's papers? So I would definitely recommend it. So Gavin Reed was basically saying that in the gas phase, that the phosphorylation can rearrange to the other site. So in the collision cell, this phosphorylation is now localizing itself to the other site, other serine. It's another whole problem. That's a challenge to work through, right? It's but there's no chance of doing this without having this retention time information. We can actually begin to get a sense of how big of an issue it is by having this data, because this retention time information means it belongs onto this molecule when it was separated from the site. So I'll point out that though Gavin Reed's papers are very controversial, despite the fact he did an enormous amount of validation to kind of show that this rearrangement was occurring. There's a lot of people that don't quite believe it, but it's definitely very clear in the data that Brian's been showing that, at least under the conditions for which we collected the data, on the instrument we collected the data, that it's definitely not as unusual as people have tried to claim it is.
Also, the paper's all about flex, so totally different instrumentation platforms, obviously.
Yeah.
QEs, so this is more widespread, I think,
Yeah.
than just us.
That's right. So here's another example. So here's if you had, so what some people have said, well, I've got the MS1 data here, right? So why don't I can use, if I can go look, for other MS1 signals the same mass, and we can integrate them as long as I had in multiple different runs, I'm able to identify them enough times, I can then eventually, figure them all out. But there are a lot of cases where they're not chromatographically resolved. And you can actually look at the product ions that are unresolved, and you can start to separate the individual positional specific fragment ions. You can still see again here some of the gas phase rearrangement occurring in that case as well. Again, so this is another interesting case. And a lot of, with DDA, people often use like a positional scoring algorithm, like A-score, like that the Gigi lab developed. One of the challenges, of course, is that if you have things by DDA, you're not going to do very well when you're not chromatographically resolving two components. So it doesn't really do very good if you have, where your two components are less than 5 seconds from one another, 15 seconds from one another, when, of course, the source, because it's collecting all the data continuously across time, can still get by with that. Okay, kind of. There's also, just want to show, there's even cases where they're not chromatographically separated at all, and functionally, whether or not you have an inhibitor with or without insulin, in this case, the phosphorylation is changing in a specific way, in a site-specific way, that would have been completely missed by another algorithm. Okay, so just in conclusions, so data-independent acquisition, obviously people have kind of drunk the Kool-Aid a little bit if you're taking a course on data-independent acquisition, offers systematic sampling over traditional discovery proteomics. We have the ability to perform a DIA with selectivity of 50% the precursor isolation window across the entire mass range using those overlapping windows. I definitely encourage people to do it. It works, and it just it's definitely, from what we can see, there's no losses. Every experiment collected in our lab uses overlapping windows. There's a Jasmus paper describing some of the details that came out this year. The dynamic range and sensitivity of our DIA approximate PRM, but it's comprehensive. So I didn't talk about this, but in cell lines, Brian's been able to show that about like 90 to 95% of the peptides he finds in the narrow window DIA can be found in the wide window DIA. So that's pretty powerful, right? So basically, we're using the extra information of that on-column chromatogram library to be able to gain back some of the selectivity that's lost from using a wide precursor isolation window. The dynamic range is, so signal can be calibrated between labs and instrument platforms using common external reference samples. I think this is something where we need to put more effort into it and think about 4 different sample types, what should be the common reference. You guys should be thinking about when you do experiments, what am I going to be making my measurements relative to? And think about that before you begin your experiment design. But as Brian pointed out, if you don't have that peptide in your common reference, you're not going to be able to calibrate it between labs or between groups. So we often try to think about these things as pools, often. We in our lab try to, I'll point out, we still do protein roll-up in some cases, but you got to be careful with doing protein roll-up. It's in almost every case that we've looked at it in detail, there's interesting things occurring on the individual peptide level that are lost when you roll them up into individual proteins. You have to be careful with it. You do gain statistical power, though, by rolling things up into proteins, right? So you gain back that other aspect. You go from 30,000 peptides to 5,000 proteins, and not only you have multiple peptide measurements for an individual protein, but the downside is that even the things like A beta, which has been known for 40 years as being important, is lost on the protein or gene level. So proteomics assays that measure lots of peptides also require lots of samples. So yeah, be careful what you wish for. Everybody wants to be able to measure more things in a single measurement, but when you do that, you have to be thinking about, is your experiment powered correctly? You're much better off figuring out which are the peptides that are precise and accurate and quantitative, and then making those measurements across many samples, as opposed to just, I've got the biggest list and the biggest library I can find, so I'm going to measure all those things that I can find that are in there. So be careful about modified variant peptide labels are not being assigned to unmodified peptides and vice versa. This is definitely an area of research still. It's definitely something that I think is going to be ongoing for a bit. Positional isomers require scoring that take into account positional information. In fact, data independent acquisition is probably one of the best ways to handle things like phosphorylation, phosphoproteomics. It hasn't quite gotten out there yet. It is controversial. Manuscript with 3 reviews, pretty harsh. We're going through. Definitely took a little bit to get it, to get through the reviewers' minds, but I do think that once more people start to do this and the tools become a little bit easier for people to use, that people will begin to appreciate that more. And I'm happy to answer any questions. I'll point out that I did none of this, so I'm fortunate to have a great group of people that we work with. You've already met, Brendan, of course, and Brian Searle collected a lot of the data within this lab. Also, Lindsay Pino contributed a lot to the information that was in the presentation. We have fantastic collaborations, including Olga's lab and Andy Hoofnagel, and we also worked closely with Bill Noble and Jake Jaffe, who's here in the Boston area. And we're fortunate to be supported financially by the federal government and also instrument vendors. And I'm happy to answer any questions. And here's a very outdated photo of the lab.
For the external single point calibration, can you comment on the accuracy of measurement in the reference sample? Because if you have to measure and analyze, not all of them are accurately measured, and then you may actually do harm.
Yeah. So what we often do is we start by taking that common reference and we make the dilution curve. We determine the limit of quant from that. We don't always do that prior to collecting our DIA data, but sometimes we do it after. And then what we often do is only look for the peptides that then are above the limit of quant and have a certain precision. At least that's the broader goal that we would have, right? So you would have, so people often talk about assay libraries, like I have a spectrum that I saw in one data set at one point in time. That's really not an assay library. What you should have as an assay library are those things that meet your criteria for accuracy, precision, dynamic range, and detection, right? So I think that's something that we would like to get to. It does require extra experiments, a little bit of forethought. So we've done those sorts of experiments in CSF and plasma, things in our labs, doing a fair amount of work in, and yeast, but we haven't done it across all of our samples that the lab's collecting. And what we warn people about is that those data should only be really thought of as being differential. And because we don't really know over what range of intensities that the experiment's linear. So I think that's, so it's nice to have a peptide measurement where you can say that if it's disappeared or it's below a certain signal, that means it's less than a certain quantity. That's what we'd like to be able to get to, but we're not quite